Welcome everyone to today's lesson on blood. This is a continuation of a previous lesson, Blood Part 1. What you can expect in this video is to wrap up what we were previously discussing about plasma proteins. Next we are going to talk about the formed elements, and so how they are made, hematopoiesis. We'll talk about thrombocytes or platelets. We'll talk about their role in hemostasis or stopping blood loss from damaged tissue. We will talk about how vitamin K and blood thinners work in conjunction with hemostasis or to prevent it. And then we will wrap up with leukocytes. In a future lesson, we will wrap up the concept of hematopoiesis, or this creation of the formed elements, by talking about the erythrocytes, or the red blood cells. So in the last lesson, we were talking about the contents of whole blood. We discussed how whole blood, when it is drawn, looks pretty consistent. It's a nice deep red, pH of about 4. When you centrifuge the whole blood with the addition of anticoagulants, and we'll get into that today, what we see is this nice separation between the plasma, what we can see here, and the formed elements. So we discussed how plasma is usually about 55% of the whole blood volume. Of that 55%, so of that fraction of the whole blood, most of that is actually water, with about 10% solutes within it. Now, for the most part, these substances here are metabolic byproducts, or they are dietary substances that purposely circulate within our blood. Hormones, of course, are generated by endocrine glands, and of course they circulate in the blood, looking to control far off organs by these chemicals binding to receptors on those target organs. Proteins, of course, have to be created in cells, and they're created specifically for the purpose of circulating in the plasma. For the most part, these proteins are created in the liver. We'll talk a lot more about that later this term, but there are a few exceptions, and they are actually made by the white blood cells. Okay. At the end of the last lesson, we were discussing how albumin is by far the most abundant plasma protein in your blood. And the reason why we have this protein, which we would normally associate with egg whites, is that it generates osmotic pressure. Essentially, it ensures that water is reabsorbed from the tissues back into the blood. The remainder of the plasma proteins can be divided into a few groups, and there's a lot of diverse functions. The next most abundant plasma protein are the group called the globulins, and about a third of the plasma proteins are globulins. There are a few different types, and you actually might even know a little bit about these already. Alpha and beta globulins play a role in the transport of lipids. Of course, lipids are hydrophobic, and so they don't really mix with the water in your blood very easily. And so in order to transport these really energy-rich, albeit hydrophobic molecules throughout the blood, so that this energy source can be delivered to the tissues that need it, is that we actually have to package them up into these nice spheres, which are, of course, largely protein in nature, and they are the alpha and beta globulins. These are two images of alpha and beta globulins, more commonly known as LDL and HDL. And what we can see is that in each of these spheres, we have housed a lot of lipids, right? So this can be cholesterol, it can be triglycerides, it can be phospholipids, all sorts of different things are housed within these LDL and HDL spheres. Now, as you can see, HDL, or high density lipoprotein, is a lot smaller than LDL is. And actually, these two types of globulins have kind of opposite functions. Okay, both of them are critical in transporting things like cholesterol around your body, but they take that cholesterol literally in opposite directions. Okay, so what we can see is that HDL down here actually takes cholesterol and other fats from your blood, so essentially from your diet, and circulating throughout your blood, and they actually take it to the liver. Okay, the liver's job, of course, is to regulate the levels of all sorts of different things in your body, so the liver can then store this extra lipid. Right, so the fat, the cholesterol, whatever, they can store it within the liver cells, therefore it's out of your blood, hooray. Or if we have plenty of these fatty substances, the liver can actually break down those fats and remove them from your body. So essentially HDL is removing these fatty substances from your blood. LDL, on the other hand, takes lipids, right, both from your diet as well as that which is stored in the liver, and it actually brings it back into the blood. So essentially LDL is taking cholesterol to the heart, to the rest of the body, and HDL is removing it. And so usually there is this balance between LDL and HDL, usually referred to as the bad cholesterol as LDL, and HDL is the quote-unquote good cholesterol. And the reason for that is, of course, that LDL is taking the cholesterol to your heart and to your arteries, and so if you have an abundance of LDL and you have a poor diet and you have a genetic predisposition to having high cholesterol, essentially this is packing your arteries full of this really sticky, globby, fatty substance, and of course that can generate plaques or atherosclerosis and can cause a lot of issues, and we'll start to talk about that today, and we'll continue to talk about that through the rest of this particular unit. Again, HDL is usually called the good cholesterol. It's not that cholesterol itself is good or bad, right? Good or evil, whatever. But HDL is actually removing that cholesterol that LDL just brought. Okay, so that's why we get this good and bad connotation. Okay. The next type of globulins are the gamma globulins, and these are actually the one exception to the proteins made in the liver rule. In fact, these gamma globulins, also called immunoglobulins, are made by the B cells or the B lymphocytes, which are a type of white blood cell. We'll talk all about them in the next unit. But essentially, these guys are super specific. They bind to a particular, what is called epitope, or essentially a little pattern on the surface of a bacteria or a virus or whatever, and they actually launch an immune response against them. And so we always have antibodies circulating within our blood. Of course, they're only there if we've been exposed to this particular substance before. And so the stars here are just a reminder that these are the one type of plasma protein which is not generated in the liver. Okay. Another very important plasma protein is called transferrin, and if we break this word down, we can see trans for like transportation, moving around, and ferrin is indicating iron. And so the deal here is that iron is critical to our existence, but not only our existence, it is critical also to invading pathogens' existence as well. And so it's really important that iron is bound to something so that, first of all, bacteria that are trying to invade your body can't get a hold of it and therefore bloom and take over our entire body. And secondly, free iron can be toxic to the human body, and so we really want to make sure that iron circulates in our blood bound to something. And so this transferrin, again made in the liver, literally transports iron around the body so that we can actually recycle the iron that is used in our red blood cells and dump it back into brand new shiny red blood cells. Okay. The final group of plasma proteins that we'll talk about today, actually quite extensively, are all involved in blood clotting. So what we will see is that proteins called fibrinogen, prothrombin, and a variety of clotting or coagulation factors always circulate within the blood in an inactive state. These clotting factors have several different names. First of all, we have a series of factors, all named with Roman numerals, that are dependent upon vitamin K in order to actually be formed, and more about that later. But essentially, prothrombin, fibrinogen, and all of these factors are circulating throughout the blood in an inactive state. That is, they are not going to lead to the formation of a blood clot. Okay. What is also circulating in the blood in an inactive state, we're going to talk about this more in a few minutes, are platelets. Okay, platelets are some of the formed elements. We tightly control how many of them we have in our blood at any given time, and these are ultimately fragments of larger cells within your bone marrow. Now, the platelets are also kept in an inactive state, but they have to be actively kept. Inactive and mobile, and essentially what happens there is that more or less there's nothing sticking out of the vessel walls in order for platelets to stick to. Pretty much anything irregular platelets are going to start attaching to it and each other. And also, the blood vessel walls are constantly secreting nitric oxide and other chemicals like prostacyclin. And essentially, these are signals that keep the platelets from sticking to each other and initiating the process of blood clotting. Now, as soon as you cut your skin, right, and blood, of course, starts pouring out from your blood vessels, what's going to happen there is that this irregularity and even loss of inhibition from the blood vessel walls is going to allow platelets to start sticking together. Right? And as soon as they start sticking together, they are actually going to turn on this series of clotting factors. What we can see here is that when platelets start to stick together, they release chemicals that initiate what is called the clotting cascade. And so this here is something that we'll talk about again in just a couple minutes. But essentially, each of these represents a clotting factor. When this one is turned on, it's going to turn on this one, which turns on this one, which turns on this, and so on and so forth. And so essentially this process gets amplified really quickly just with that initial turning on of the platelets in the beginning. And we'll watch an animation here in just a few minutes that essentially makes this process out to be kind of like a big pinball machine, and it really is that fast. One hits another, hits another, hits another, and the response is amplified. And so this is a great thing. This clots your blood right away to prevent any further blood loss. And so this is an image that we are going to be walking through. Here's the damaged blood, or here's the damaged blood vessel, you're bleeding, all of a sudden platelets are going to stick together, these clotting factors and other inactive proteins are circulating in your blood, they're going to be turned on, and essentially you are going to form a nice scab that prevents any further blood loss. And so this entire pinball-like process is called a clotting cascade. Okay. And so what stimulates this? Well, essentially any irregularity in a blood vessel wall. Okay, so when you cut your skin, of course, there is damaged tissue, and so platelets are going to start sticking together. What also might stimulate this clotting cascade is any kind of irregularity in the blood vessel wall, such as a plaque. Remember a few slides ago we talked about how LDL is going to bring cholesterol and such towards your heart, and so if you have too high of a quote-unquote bad cholesterol, that can lead to the formation of these fatty plaques within your blood vessels. And so this provides an irregular surface to which platelets can bind and therefore initiate this entire clotting cascade. All right, so that is obviously not an ideal circumstance. And again, damage to the vessel wall. And finally, anytime your blood is not in your body, right? Anytime it's not circulating within these blood vessels, it's not getting those signals to turn the platelets off. Right, so remember that the linings of these blood vessels are always releasing nitric oxide and prostacyclin and other chemicals to keep the platelets less sticky. And so when you remove blood from the body, they're no longer the platelets are no longer getting those signals. And so blood that is removed from the body automatically clots, right, forms clots. And so that has implications for the removal of blood from the body. Okay. In the first part of this blood lesson, we talked about plasma versus serum, and I said that we would come back to that, and now we finally have enough information to be able to understand the difference between serum and plasma. Okay. Remember that plasma is the fluid in your blood that contains all of those clotting proteins. Remember that clotting proteins are going to spontaneously form clots in the blood unless they are told otherwise, right? Unless nitric oxide, prostacyclin, etc. is keeping them from turning on. And so when you are administering plasma or whole blood to a patient via an IV line, essentially there has to be a tiny bit of an anticoagulant within that blood to keep it from spontaneously forming clots and therefore giving clots to the patient receiving a blood transfusion, right? Obviously that is not an ideal circumstance. Okay, if clots are allowed to form in a blood sample, right? Obviously you would not be giving this to a patient, but if you draw blood and you do not treat that blood with some sort of anticoagulant, with some sort of chemical that prevents the formation of clots, essentially what's going to happen is that all of those clotting proteins within the plasma are going to automatically form a clot with the platelets, which we know are in the buffy coat. And so what you're left with in that case, if you don't treat with anticoagulants, is a big old blood clot. Okay, and of course, this often includes the red blood cells as well. And the fluid that is left over no longer contains those clotting proteins. Those clotting proteins are already down here in the clot. Okay, so the fluid left over is called serum. So serum is essentially the plasma, but without those clotting factors, without those extra proteins within it. Okay, so before we move into the next topic here, I do want to take a brief moment to just go over a couple practice questions with you. First, what is the most abundant liver protein in the blood? Okay, immunoglobulins. These are antibodies, and in a general healthy individual, they are not the most abundant liver protein. Transferrin transfers iron throughout the body, helping us to recycle it in the life cycle of the red blood cells, not the most abundant liver protein. Okay. LDL is transporting cholesterol, and we certainly hope that this is not the most abundant liver protein. Albumin is about 60% of the plasma proteins, and so that is indeed the correct answer. And finally, fibrinogen. This is one of those inactive proteins that is involved in the clotting process. However, it is not the most abundant liver protein. So the answer here is D, albumin. And I do want to make one more point here. I specify liver protein. Remember that the liver is not responsible for making immunoglobulins. These are made by a type of white blood cell called the B lymphocytes. Okay. One more practice. Which of the following is a correct statement about whole blood? The ideal pH is 7. This is not true. The ideal pH is 7.4, although generally it remains within the 7.35 to 7.45 range. It contains about 80% water. This is also not correct. Remember that whole blood, on average, contains about 55% plasma. Plasma is about 90% water. Okay, so our blood in total does not have 80% water. All right, next, C. It contains about 45% leukocytes. This is not the case. This would be very bad. It does, however, on average, contain about 45% erythrocytes. Okay, so we have about 45% hematocrit, or red blood cell content. Leukocytes are less than 1% of the whole blood volume. Okay, moving on, D, the total volume is about 10% of your body weight. This is also not correct. The total volume of your blood is about 6 to 8% of your body weight, which generally works out to be about 4 to 6 liters, of course, depending on how large of a person you are. Okay, which hopefully means that E here is the correct answer. Whole blood contains a relatively higher concentration of sodium than it does potassium, and yes, indeed, this is correct. Remember that these concentration gradients are maintained by the sodium-potassium pumps, and this is critical to the formation of action potentials, muscle contraction, and many other functions that we will learn about this semester. Okay, so that is all for plasma. Okay, remember plasma differs from serum in that it contains those clotting proteins. Okay, next let's move down to the formed elements. Remember that when whole blood is treated with an anticoagulant such as heparin or warfarin, and we centrifuge that blood down, what we get is this nice separation between the formed elements and the plasma up here. The heaviest of all the formed elements are the red blood cells. Right, so they go all the way to the bottom. We can also call this percentage of red blood cells the hematocrit. Okay. In between the plasma and the red blood cells, we have what is called the buffy coat, and this is less than 1% of your blood, and it contains both white blood cells and platelets. Now, in general, all of the formed elements are formed in the same place. Remember that all of these substances, white blood cells, red blood cells, and platelets, are all tightly regulated. We control how many of them are in the blood at any given point, and they are purposely put in the blood. All of the other things end up there, but the formed elements are literally formed for the blood and the blood alone. Okay. The process of developing all of the formed elements in general is called hematopoiesis. Okay. Hematopoiesis in an adult occurs in the bone marrow. However, if we look at this graph at the bottom, we can see that the location of most of hematopoiesis is going to change over the course of your development and over the course of your life. And so I first want to point out what's going on when you are a fetus. All right, so here's conception, here's 0, and of course birth over here. Now, as we can see, the bone marrow is not the location of hematopoiesis until after the 4th month, and perhaps this makes sense. You don't have developed bones really until after this particular time. And so before you have bones that have a distinct spongy bone versus compact bone, essentially all of your formed elements are going to be created somewhere else. And even that changes. First of all, before you even have organs, the yolk sac, yes, humans have a yolk sac. We'll get into that at the end of the semester, but the yolk sac is actually responsible for making your formed elements. Okay, some of the first organs to really start to develop are the liver and the spleen. Okay, and so both of these organs are going to slowly take over the process of hematopoiesis or formed element formation. Okay. We can see that the liver and the spleen become less important for this process as the bones become more and more developed. Okay, so by the time you're born, your bones, right, your bone marrow has fully taken over this process. Now, I do want to point out that the lymph nodes are also going to be important in part of hematopoiesis for the rest of your life. Okay, we'll talk about this more in the next section of this class, but essentially white blood cells kind of hang out in your lymph nodes, and if anything that doesn't belong ends up in your lymph nodes, the white blood cells are going to undergo mitosis after mitosis after mitosis, and they're going to make so many more of themselves and ultimately dump them into the blood to launch a full-on body-wide immune response. Okay, so that's what's happening in the lymph nodes. Only certain types of white blood cells are going to replicate there. Okay, but they do have to start in the bone marrow ultimately, right? And so we'll get into that later this semester. Let's go back to the bone marrow. We can see that the bones are sustaining the body's needs for formed elements by the time you're born. But we do see that the relative importance of each of these different bones is going to shift a little bit over the course of your life. When you are actively growing, okay, so as a child through adolescence, you are growing a lot, and as a result, you need a lot of blood, right? You need a lot of energy transported around your body, and so you are continually making more and more and more of these blood cells. And so during your childhood, your tibia and your femur, right, so your leg bones, are super important in this process, okay? However, as your growth slows down, hematopoiesis is similarly going to slow down in these bones. Okay, so when you are an adult into old age, the primary locations of hematopoiesis are in your vertebrae, in your sternum, in your ribs, and of course in your pelvis. And of course, if you need a bone marrow transplant, or donating bone marrow, generally they take bone marrow from your hips, right? And the reason for that is that the sternum is super close to all sorts of organs that we desperately need. If the physician makes a mistake in extracting the bone marrow, essentially it can get pretty bad pretty fast, right? If you accidentally poke your heart instead. And so the pelvis is relatively isolated. There's a lot of muscle around these bones. And so if there are any mistakes, it's not a lethal mistake. Okay, and just before we move on to the next slide, I want to remind you that bone marrow is housed within the spongy bone of any of the bones that we just talked about, right? So this one happens to be the femur, and so in the epiphyses we see lots of this like spongy stuff. That's the spongy bone, and within the gaps between the trabeculae, that is where the bone marrow is located, and so that is where hematopoiesis is occurring. Okay. This chart is something that we'll refer back to a couple times, but I want to point it out now because all of these different formed elements, okay, so the red blood cells, the platelets, and all of the lovely different types of white blood cells all derive from the same type of stem cell. Okay, so this is kind of the parent cell of all of these different formed elements. We call them hemocytoblasts, right? Hemoblood, cytocell, blast essentially means like a building cell, and so these are really like the building blocks of all of these formed elements. We can also call them hematopoietic stem cells, and so these are multipotent stem cells, which essentially means that they have not differentiated very far yet. That is, they can ultimately divide and become any of the formed elements that we see down here. Okay, so they are multipotent. They can become multiple things ultimately. Every time we see one of these divisions, what that's indicating is that a stem cell has undergone mitosis. Right? And the daughter cells, or one of the daughter cells, differentiates, right? That is, it turns on certain genes, it turns off certain genes, and this is an irreversible process, which means that these cells, right, cells that grow up to become myeloid progenitor cells, can only ultimately produce the thrombocytes, the macrophages, the dendritic cells, right, and all of these guys. They cannot produce lymphocytes. They cannot produce natural killer cells. Okay, so once one of these like branching events has taken place, that indicates that the cells farther on down the line are all going to be derived from this cell, right? But these cells and all of their offspring ultimately cannot become any of those cells over there. Okay, and so it's just different levels of what is called differentiation. You cannot turn back the clock and, become one of these hemocytoblasts again. Okay. Ultimately, what kind of pushes this differentiation, right, into a myeloid versus lymphoid progenitor cell are hormones, right? So depending on what you need, right, if you need more red blood cells, you'll have one hormone controlling the process. If you need more T lymphocytes because you are, trying to fight an infection, you will have different hormones or growth factors, right? And ultimately, this will drive the developmental process in either direction. Okay. Once again, blast in general means building up. So the B and the B, the blast is the building cells. These are the building blocks, or in other cases they build bone or they build fibers, etc. Okay, so let us look back just a little bit to where this is occurring. Remember that hematopoiesis, right? All of those different cells are born in the same spot from those hemocytoblasts, those multipotent stem cells. Okay, and all this is occurring within the spongy bone here. After differentiation into, an erythrocyte, into a monocyte, whatever the cells are going to go up to be, after that, these cells, of course, have to end up in the blood somehow. And so they're going to enter the blood through these really special types of blood vessels. These blood vessels are called sinusoid or discontinuous capillaries. And actually, we'll talk more about the different types of blood vessels in our next lesson. But for now, I want you to see that normally your blood vessels are pretty darn solid. Formed elements are, for the most part, supposed to stay inside your blood vessels, right? It only works if it's inside your blood vessels. And so, in order for these larger formed elements to actually cross over a blood vessel wall, we need super porous, super leaky almost blood vessels. And see, these blood vessels are called sinusoid capillaries. We can see that the cells lining these capillaries actually have big old gaps in between them. And so these gaps are big enough, as we can see over here in the histo image. These cells are actually big enough to cross through those big gaps and enter into the bloodstream. Okay. All right, so once again, I do want to remind you that all of the formed elements ultimately are born in this way. They are ultimately originating in the bone marrow. They enter into the bloodstream via these sinusoid capillaries. However, some formed elements, namely the white blood cells, have to continue the developmental process somewhere else. And again, that's something we'll get into when we talk about the immune system in a few weeks. Okay, so let's start focusing in on hematopoiesis by following the development of thrombocytes, that is, following the development of these platelets. Remember that all of these formed elements are derived from these stem cells. Right? Some hormone growth factor is going to cause the daughter cells of these stem cells to differentiate into a myeloid progenitor cell. Okay, now these myeloid cells can still become all sorts of different things. Okay, so they're going to undergo mitosis after mitosis after mitosis, and some of those daughter cells are ultimately going to become megakaryocytes. Okay, and so what is actually going to push the daughter myeloid progenitor cells to become megakaryocytes is a chemical called TPO, or thrombopoietin. Okay, once again, this chemical, which is produced mostly by the liver and to some extent by the kidney and the bone marrow, so a lot of different organs can actually produce TPO, but essentially when it binds to receptors on the surface of a myeloid cell, it is going to trigger that cell to produce megakaryocytes. Okay, and so really what are megakaryocytes? These cells, as you can see, are like big and clunky. They have all sorts of nuclei. They have all sorts of like stuff floating around in their cytoplasm. So essentially these daughter cells are going to undergo mitosis after mitosis after mitosis. However, those daughter cells are never actually going to divide, right? So they never actually undergo cytokinesis. And so what you end up with is this big, clunky, multinucleate cell with all sorts of really cool things in the cytoplasm. Okay, this image here is showing us the bone marrow up here. So this is where those megakaryocytes are first produced. And these blood vessels are sinusoid. So this is how all of the formed elements are going to get into the blood from the bone marrow. And so the way that we go from megakaryocyte, big clunky multinucleate cell, to these tiny little platelet fragments of cells is that the megakaryocytes actually extend a piece of their cytoplasm into the vessel. So ultimately, the shear flow of the blood is actually going to break off tiny little pieces. And so these pieces of megakaryocyte happen to have inside any of the chemicals, any of the vesicles that were present within the megakaryocytes. And so platelets are essentially little pockets of all sorts of different chemicals. These chemicals, as you might imagine, are all involved in the clotting pathway. And so we'll talk about that in just a moment. Before we do, I also want to point out that these platelets contain actin and myosin, and so you might recognize these terms as being part of our skeletal muscles, and in fact our smooth muscle and cardiac muscle as well. This is what allows stuff to contract, and so platelets are going to be really important in clotting and healing tissue, and they're actually going to use actin and myosin to kind of pull the edges of your cut closer together. So kind of cool. Stay tuned to later in this lesson. Platelets constantly are being made and broken down. Now keep in mind that these are anucleate, there's no nucleus in them, and so if they get damaged in any way, which inevitably that happens, there's no way to fix themselves. And so these platelets actually break down after a week to a week and a half. And so megakaryocytes are always made, and those megakaryocytes are always making more platelets. Okay. And so at this point, we have enough information to talk about how we stop the loss of blood. Okay, this process, so the clotting process itself, is called hemostasis. And I know that this sounds like homeostasis, and yes, hemostasis is required to maintain homeostasis, but please make sure that you get these terms straight, right? Hemo means blood, stasis means stop, and so hemostasis means the process of stopping the blood. Okay. Generally, this process immediately stops the loss of blood when you damage your tissue, and it's actually going to start the process of fixing your tissue right away. All right, so we'll talk about that in just a moment. For the most part, textbooks will divide this process into 3 different phases, but keep in mind that it's not like step 1 happens, that finishes, step 2 happens, that finishes, step 3 happens. Instead, all of these things are kind of overlapping and kind of feeding off of each other. So for simplicity, we are going to break it down into 3 steps, but again, keep in mind it's a little bit more complicated than that. Okay? In order to introduce the process, at this point, let's watch a short animation. Keep in mind that there are a lot more words in this animation than you actually need to know. So we're going to watch the animation, and then we're going to come back together, and I'm going to walk you through what you need to know.
Normally, the flow of blood through blood vessels is unimpeded. Red cells, white cells, and platelets pass over the unbroken lining of endothelial cells. Its smoothness discourages their adhesion. But there exists, within this supporting framework of proteins and mucopolysaccharides, an emergency mechanism capable of reducing blood loss by promoting coagulation. This is the extracellular matrix, or ECM. The first reaction to an injury involving the epithelium comes from the damaged epithelial cells themselves. They release endothelin, a polypeptide which causes smooth muscle in the wall of the artery to contract, as do nerve impulses arriving in response to the injury. As a result, less blood is lost because the vessel is narrowed at the site of injury. The exposed ECM releases von Willebrand factor. This attracts platelets to the damaged area and promotes adhesion between these platelets. It forms bridges between platelets and the underlying extracellular matrix. Von Willebrand factor is also found on the surface of epithelial cells and in platelets as alpha granules. As platelets adhere to one another and to the extracellular matrix thanks to von Willebrand factor, they become activated, causing a reorganization of their actin fibers and a change in their shape. This in turn exposes their lipoprotein surface to the blood clotting factors. The activated platelets now release adenosine diphosphate, or ADP, and increase the synthesis of thromboxane A2, both of which boost platelet attraction. Both of these molecules attract more platelets to the injury site, expanding the platelet mass and further plugging the breach. Secondary hemostasis is the process of stabilizing the clot. It begins with the coagulation cascade, a sequence of events which develops with explosive speed, ending in the formation of a fibrin clot. It starts when factor VII binds to tissue factor. This activated complex attaches itself to the platelet surface, setting the cascade in motion. It activates factor X so that there is a rapid increase in the level of activated factor X, 10A, in the blood. Activated factor X cleaves prothrombin, yielding thrombin. In turn, thrombin cleaves fibrinogen to form strands of fibrin. We have been tracing the rapid response pathway of clot formation, from injury to the cells lining blood vessels through to the formation of the fibrin network. This pathway is known as the extrinsic system. But there is another, slower pathway called the intrinsic system, which may be triggered by exposure of blood to a negatively charged surface, such as collagen or glass. The two systems are indeed complementary pathways which join at the level of activation of factor X. Polymerized fibrin forms a network which anchors blood cells and platelets in the clot.
Okay, so the video itself didn't break down hemostasis into these three phases, but let's add them on because this is what every single textbook is going to tell you about hemostasis, or the stopping of blood. The very first thing that happens is what is called a vascular spasm. Okay, so this is the phase that starts seconds after the injury and lasts for about a half an hour after the injury. Essentially what's happening here is that when the tissue is cut, what's going to occur is that first of all, those endothelial cells, those lining cells of your blood vessel, are going to release chemicals that are essentially signals that say, oh my gosh, like, we have a problem here, we need to fix it immediately. And so one of the primary chemicals here is called endothelin. Notice that this chemical is in blue and underlined, indicating that this is one of those words that you need to know, right? You don't need to know von Willebrand factor and all that other kind of stuff, but endothelin is one that you should be familiar with. And so essentially, endothelin, as well as some nervous system feedback, causes the smooth muscle lining your blood vessels to contract. And so this leads to something called vasoconstriction. The wide blood vessel previously wide blood vessel is going to constrict. Right, so the diameter gets smaller, and ultimately this allows less blood to the site of the injury, and therefore we lose less blood to the injury. Okay. Also, this chemical is going to begin signaling the remaining blood vessel wall cells, those endothelial cells, to start the process of mitosis, right? So when you cut yourself, there's actually like a hole between those cells, and so we need to make more cells as quickly as we possibly can to literally fill in that hole. What's also happening is that when this tissue is damaged, right, it exposes collagen. Right, so collagen is just naturally part of the extracellular matrix, and so when the tissue is disturbed in such a way, the collagen becomes disorganized, and we can actually see that in this diagram that I drew over. The collagen fibers start sticking into the lumen of the blood vessel, and as we talked about earlier in this PowerPoint, platelets will automatically stick to stuff unless their environment is perfectly smooth. And so this exposed collagen is automatically going to attract the platelets. And so as soon as the platelets start to attach to the collagen, they're also going to start attaching to each other. Okay, and so this is going to begin hemostasis phase 2. Now the vascular spasm is still happening, but now we are beginning the platelet plug phase simultaneously. And so this is one of the great examples in our body of positive feedback. This is an emergency situation. We need to plug the hole, stop the blood from leaving our blood vessels. And so as soon as some of these platelets stick to the exposed collagen or to each other, they are going to start releasing their own chemicals. Remember that platelets are chock full of whatever was in the megakaryocytes. Here we get to actually see what was in those megakaryocytes. Okay, so they're going to release ADP, as the video said. Okay, ADP automatically stimulates more platelets to arrive at the scene and to start sticking to each other. Okay, and again, once a platelet sticks to something, it releases more ADP. More ADP causes more platelets to come over and stick to each other, and so ultimately this is going to intensify the process quite significantly, also quite rapidly. Okay. What's also happening here is that the platelets are going to release platelet factors. Okay, so these play a role in clotting. They are going to help to stimulate that clotting cascade. The platelets aren't just there to, kind of slap a band-aid on the problem right this second. They're also there to kind of begin the process of healing right away within minutes of the wound occurring. And so they're going to release PDGF, platelet-derived growth factor, and so this is going to bind to the neighboring cells, and it is going to stimulate the process of mitosis. Again, we need to fill in that hole that was created by the damage. Finally, platelets are going to release calcium ions. Remember that calcium causes stuff to bind to other stuff, so the more calcium there is in the environment, this is going to speed up platelet aggregation, or platelets kind of sticking together. Also, calcium is really important in muscle contraction. So remember that these platelets contain actin and myosin. So with calcium around, what's going to happen is that you're going to see the edges of the wound starting to be pulled together. Okay, so calcium is really important in that process. And so before we move on to the next slide, I do want to point out that we've all kind of seen this platelet plug before, I'm sure. So when you cut yourself, between the time when blood is pouring forth from the wound and you actually develop a scab, there's that like little gummy thing in the wound. If you wash the wound after that, this thing just like pops right out and you start bleeding again. So you actually have to wait for that gummy thing to actually turn into a scab, right? Y'all know what I'm talking about. That gummy part is a platelet plug, and so it is not a scab, it is not a clot just yet, because it is still soluble in water. It can still be washed away or, bumped out of the wound, but it does set up the framework for the next step. And so the video called this next step secondary hemostasis. This is actually the process of forming a legit clot. Right? This is also called coagulation. Right? So forming of a blood clot, not just platelets sticking together, not red blood cells sticking together. We'll talk about that at another time. This is true coagulation. And so this process starts about 30 seconds after the wound has occurred. So again, this isn't step 1, step 2, step 3. This is all kind of happening simultaneously. But in order for the coagulation phase to proceed, we need the vascular spasm and the platelet plug to begin. Okay, so the goal of the coagulation phase is essentially to solidify that platelet plug, right? We want a true scab. We don't want the platelet plug to be the permanent solution to this injury. And so the coagulation phase includes the clotting cascade, right? As soon as the platelets start releasing these chemicals, it turns on all of those clotting factors. One turns on another, turns on another, turns on another. Okay, at the end of that big convoluted clotting cascade that you are by all means not responsible for learning, ultimately, at the very end, we get the conversion of what is called prothrombin, right? One of those liver proteins circulating inactive in the blood, and it is then turned into thrombin. Okay, so this is an active protein, again, turned on by the clotting cascade. Okay, and just as a reminder, this is what that clotting cascade looks like. Again, you do not have to memorize this, and you don't even have to differentiate between the intrinsic and the extrinsic pathways. Okay, so ultimately, the thrombin protein happens to be an enzyme, and this enzyme converts yet another one of these fancy proteins, fibrinogen, into fibrin, and fibrin really is the key here. Fibrin is what forms the mesh, forms the network to actually strengthen the platelet plug, right? So it actually like secures red blood cells, white blood cells, platelets, all of these different things that we're trying to get out of the blood vessel. It secures them together and actually forms something that's hard and something that is insoluble. So even when you wash your wound later, the scab doesn't just pop out. Okay, and so at this time, we also see that the platelets, which of course are surrounded by calcium that they themselves have released, we see that the platelets with their actinomycin start to pull the edges of the wound and the clot within it closer and closer together. So this really is the beginning of the healing process as well. Okay. So this again is the coagulation phase. It is the formation of a clot, which is also leading into the healing process, which we will get into in a bit here. In fact, the next time you get a wound, or maybe you have even seen this before, if you take a look at your scab, you'll actually see that it looks like the skin on either side of that wound is like a little bit tight. You see kind of like little lines going to the wound. That is an indication that your platelets are pulling the surrounding skin closer together to fix the problem as soon as possible. Okay, and so this coagulation phase should continue for about a half an hour to an hour after the wound, right? Usually at that point you formed a scab and your life goes on as usual. Okay. At this point, I do want to give you the opportunity to hear about hemostasis, right, stopping blood loss, and vitamin K, which, if you remember, those clotting factors depend on vitamin K to actually be made, and how all of this relates to what are called blood thinners. Okay, so at this point you get to watch a video from another professor that will explain how all of these factors work together.
This is Dr. David Johnson at Quillin College of Medicine, East Tennessee State University, and this talk video is about warfarin and blood coagulation. Blood coagulation involves a lot of factors that are made by the liver, and those are transported out in the blood and function out there. The big one is thrombin, because thrombin converts fibrinogen to fibrin. Fibrinogen is a soluble blood protein. However, fibrin is an insoluble and plugs up the clot. So when you cut your finger, blood clotting is good. However, if we have clots in our arterioles, that can be bad, especially if it results in a heart attack or strokes. That can damage our hearts or damage our brains. Warfarin slows that process by inhibiting the function of vitamin K. And therefore, those clotting factors, remember, are made as liver proteins, made in the liver, and then gets transported out into the blood. Vitamin K is required for a number of these that have spinning circles, and those are so warfarin will slow the function of those, make them inactive. They don't work as well, and that's measured by prothrombin time. Also, heparin is used as an anticoagulant. However, heparin has to be injected, whereas warfarin is a pill that can be eaten. And heparin is measured by the partial thromboplastin time. Vitamin K was discovered by Henrik Dam, a Danish scientist, that he termed it coagulation's vitamin, and he spelled it with a K because the Germans and the Scandinavians spell coagulation with a K. Here is the structure of vitamin K. Now, at the University of Wisconsin, the scientists there were asked to try to figure out why cows were dying when they were being dehorned. Wisconsin have a lot of dairy cattle, and so they dehorn them so they don't injure each other. And those cows were dying, and so they wanted to know why. So the scientists started looking at what the cows were eating. They found out that they were eating some spoiled sweet clover, and that spoiled sweet clover contained this compound called dicumarol, which is very similar in structure to vitamin K. So knowing that chemistry, they figured out why these cows were dying, because their vitamin K wasn't working well. The Wisconsin scientists then developed this compound called warfarin, also known as Coumadin. And warfarin stands for WARF, Wisconsin Alumni Research Foundation, and the University of Wisconsin benefited from the patent of warfarin for a number of years that's used to treat patients, and it's an excellent rat poison. The rats tend to eat large amounts of it, and they bleed to death or hemorrhage to death. Vitamin K involves a change in the structure of the protein by taking a glutamic acid residue and putting another carboxyl group on it to make gamma carboxyglutamate. Once that occurs, calcium can bind, and then the calcium allows the organization of the clotting factors in contact with negative phospholipids that are exposed when tissues are cut or membranes are disrupted. Now, this process requires a carboxylase and the CO2, and it requires reduced vitamin K. In the process of doing this reaction, vitamin K is oxidized. The oxidized vitamin K is no longer functional and has to be reduced, and there's a reductase in our bodies that does that to recycle our vitamin K. And warfarin inhibits that process, so it slows the amount of available vitamin K that's available for this reaction and slows the formation of the functional blood clotting factors. This process is measured by a term called International Normalized Ratio, and it is the ratio of our patient's prothrombin time divided by a control prothrombin time. Normally we have an INR of about 0.9 to 1.3, and in this area we can form clots pretty easily, so if we cut our finger everything's fine. However, if a patient has had a heart attack or stroke, doctors want to push their INR up, so the chances of them having another heart attack or another stroke is greatly reduced. However, you don't want to push it up too high or they start bleeding. And so we shoot for an INR of about 2.5 to 4, and that works quite well. And remember, that's measured by the prothrombin time. Here's a little device that can now be used to measure prothrombin time right in the hospital or at home, and it shows the INR and prothrombin time. It just takes a drop of blood. Once a patient is given warfarin, it takes a while to adjust the dose up and get it steady. And some people have a very fast enzymes that metabolize their warfarin in their liver, and those are called P450 enzymes, and therefore their effective dose is very difficult for them to get an effective dose, so they have to be given more warfarin. However, some people also have very slow P450 enzymes, and they can be given too much warfarin. So they need to have their warfarin levels reduced. That's why it has to be measured. The INRs are measured on a regular basis on people that are taking warfarin. Warfarin pharmacogenomics. Pharmacogenomics is the interaction of genes and drugs, and cytochrome P450 enzymes metabolize warfarin in our livers. Genetic polymorphisms in those P450 enzymes, those are all called CYPs or cytochrome Ps, such as 2C9 and 4F2, are involved in this. And there are single nucleotide polymorphisms in those SNPs, in those cytochromes that are called SNPs. And those result in a single DNA change, base change, that results in a single amino acid change, but that can make the cytochrome P450 inactive or make it function much more slowly. These can now easily be determined on chips. We see advertisements for 23andMe and Ancestry.com. These are companies that provide thousands and thousands of information on our SNPs that make us different and tell us about our ancestry and how we're related to different people. So that it's cheap and easy to get done, only about $200 to do this. SNPs used by warfarindosing.org. Doctors can use this little algorithm at warfarindosing.org to calculate the dosage. Warfarin, remember, is a antagonist of vitamin K. So leafy greens such as spinach, kale, and broccoli are rich in vitamin K. Vitamin K is fat soluble. Therefore, oily salad dressings can increase vitamin K uptake, or adding butter on your broccoli. Spinach with oil, such as olive oil, will increase vitamin K up to about 10-fold, and therefore the person will need more warfarin. Intestinal E. coli also make vitamin K, and antibiotics can reduce the amount of E. coli and therefore reduce our vitamin K, and therefore less warfarin is needed. If the INR gets too high, we tend to bleed. Vitamin K capsules are given in the liver. However, the liver makes our new vitamin K protein slowly, and so it takes a little while to adjust it back down, but it can be adjusted.
Okay, so now that we know a little bit more about vitamin K, blood thinners, and hemostasis, let's talk about getting rid of that scab, okay, or getting rid of that clot. Of course, we don't want to keep a scab for the rest of our lives, right? Our bodies will be just covered in scabs from the different injuries over the course of time. And so, what we get when a scab is formed is the beginning of clot retraction, so breaking down that clot as well. And so we know that the edges of the tissue and the vessels are pulled together by the platelets. We know that there is a lot of mitosis occurring in the surrounding cells, the ones that are still healthy, the ones that are stimulated to replicate by platelet-derived growth factor. But in order for true healing to occur, we need to get rid of this scab in the first place. And so during the repair process, the clot is literally dissolved, and this process is called fibrinolysis. So it's literally lysis, or like the exploding, the breaking down of fibrin, right? Fibrin is what actually makes the clot, and it's what we need to get rid of the clot. And so, actually kind of a wild process here. Plasminogen is activated into the, or sorry, let me rewind here for a second. Plasminogen is yet another one of these inactive circulating proteins within the plasma. Plasminogen is activated into the active protein called plasma by thrombin, right? So thrombin isn't just making the clot, right, making fibrin, it is also facilitating the breakdown process as well. So plasmin's job is to go in here and start cutting apart this fibrin and breaking down that clot, right? So again, thrombin, right, thrombo means clot, thrombin is both pro-clot and anti-clot. Right, and so plasmin is breaking down the clot while mitosis is happening in the healthy cells, and the edges of the wound are being pulled back together. Okay, so in general, these processes, hemostasis and fibrinolysis, are a good thing, right? We need to stop blood from leaving our body when we get a wound, and also we need to get rid of clots sometimes as well. And so usually we have this balance of hemostasis and fibrinolysis, and as long as this balance is maintained, we have homeostasis, right? This healthy set of internal conditions. And so this here is representing a balance, right? And surely you have seen these before in textbooks. When the balance is flat, that means homeostasis is maintained. But what happens when there's too much clotting, right? So when hemostasis outweighs the fibrinolysis, this can become a clinical issue in a lot of different ways. For example, a deep vein thrombosis, or a DVT, is essentially too much clotting in the veins of your extremities. And so this can happen well, it happens in a variety of ways, but in general, if you are not moving your muscles very much, essentially your low pressure venous return back to your heart is going to essentially allow blood to pool around your valves, right? And we're going to get into this more as we go along here throughout this unit in this class. But in general, without moving your muscles, essentially your muscles aren't helping the blood to return to your heart, so it's just kind of like hanging out in your extremities. When it hangs out too long, platelets automatically start sticking together, and as a result, they can start to form clots, right, in these extremities. And so this is a problem, of course, for circulation in your extremities, but also sometimes these clots can break free, or fragments of these clots can break free and start circulating in your blood. And so these can end up in all sorts of dangerous places, particularly when your blood vessels start to get narrower and narrower to go into smaller vessels like those in your brain, for example, like those feeding your heart muscle, for example. And essentially when these clot fragments get to these smaller vessels, they can become lodged in those vessels, and ultimately blood flow is reduced or even stopped entirely. And so that's a problem, because anything kind of downstream of that that clot is not going to be fed with blood, and therefore the tissue can die. And so this is called an ischemic stroke in this particular example. Similar concept, right? A dislodged blood clot can get trapped in all sorts of different smaller and smaller vessels, including in your lungs as well. And so when a clot blocks blood flow in your lungs, it is called a pulmonary embolism, right? And so of course, all of the oxygen that you are taking into this entire half a lung is not actually getting into your blood, and so you are going to not have enough oxygen to feed the rest of the tissues in your body. Okay, what about the other way, right? What happens if there's too little clotting, right? Again, so the balance going the opposite direction. There's too little clotting, essentially you can't stop the blood flow, right? And so an example of this, as we can see it, in all of these different photos here, is hemophilia. And so hemophilia is most commonly a genetic condition. It is the most commonly known is a sex-linked disorder. That is, there's a mutation for the clotting factor 8 gene that we can see right here, specifically on the X chromosome, and so this has a lot of implications. First of all, without clotting factor 8, we don't get clotting factor number 10, which means that we don't get prothrombin, and without prothrombin, we don't get fibrin, and without fibrin, we don't get a clot, right? So anytime you cut yourself, right, a hemophiliac cuts themselves, they just can't stop the bleeding, right? Because you can never really get past the platelet plug phase. Okay, so that's the first implication. The second implication is that this is on the X chromosome, which means that if you are a male and you inherit this particular mutation, ultimately that's the only one you get, and so you are stuck with this mutated, inefficient, or completely defective clotting factor 8. If you are a female, you of course get 2 of these, 2 alleles for this particular clotting factor, and so if one of those alleles is good, then you can still clot your blood, right? You'll just be a carrier for that particular condition. Okay, and so this disease was actually kind of passed around a lot in the European royal families a couple hundred years ago, because there's a lot of inbreeding, and so a lot of males in particular died of hemophilia because, of course, they get the hemophilia gene. The females, of course, got 2 different alleles, and even if one was a hemophilia allele, they could still use the healthy dominant allele. Okay, the final topic I want to very briefly mention for you today is leukapheresis. So this is the process of making white blood cells. Now, white blood cells, as you see in lab, have nuclei, in fact, very variable nuclei, and so because of these nuclei, white blood cells can fix the damage they accrue by circulating throughout the blood and throughout our tissues. As a result, white blood cells can live for days to decades, and so this process isn't, kind of constant, just like making red blood cells and platelets is going to be. Instead, the creation of white blood cells is a lot more tied to what you are experiencing in terms of invading pathogens. That is, anytime your body is invaded by a bacteria, a virus, a fungus, funky proteins, like whatever doesn't quite look right, it doesn't quite look like what our body should, there are specialized cells that notice that this is a problem, right? And ultimately, they release chemicals, right? And each pathogen has a different type of chemical associated with it. Essentially, these messages are specific to what is going on with your body. In general, all of these chemicals are within a class called cytokines. Okay, more specifically, the interleukins. Right? So the interleukins are a type of cytokine that can actually determine which specific white blood cells are going to be created, right? For this, we need neutrophils. For that, we need, this type of B lymphocyte, etc. Okay, and so the cells that are on the scene, and even some that are within the bone marrow and within the endothelium, for example, they release different types of interleukins. In this example, interleukin 8 is released, and that's going to do a couple things. First of all, it can actually attract certain white blood cells to the scene of infection. Right? White blood cells are kind of cool like that. They can actually wiggle out of your blood vessels and actually attack invading pathogens. And secondly, these interleukins can actually stimulate the production of very particular white blood cells. So here we can see they stimulate leukocytic progenitor cells to actually proliferate, so undergo mitosis, and differentiate into exactly what we need for this specific type of pathogen. What we can see here again is that that chart of the development of all of the different formed elements. And the reason I bring it up now is to show you that if there's any mutation in this leukapheresis process, so the creation of white blood cell process, this can lead to leukemia. And remember that emia means in the blood, leuk means white, so too many white blood cells in the blood. And so there are many different types of leukemia. We're not really going to get into the details of this, but I do want to point out that the words, like the classifications of leukemias, can be tied back to this chart, right? So there can be lymphocytic leukemia that essentially indicates that there is a mutation in one of these lymphoid progenitor cells. And so ultimately all of the cells downstream, right, the T lymphocytes, the B lymphocytes, etc., are all going to contain these mutations. Okay. Myelocytic leukemia can be a mutation of the myeloid progenitor cell, or there can actually be mutations, farther on down the line. Essentially, acute leukemia is very fatal very quickly, and so this generally involves mutations in cells farther up the line, right? So in a myeloblast cell or even within the myeloid progenitor cells, essentially a mutation in these cells that are kind of farther up the stem cell line means that those mutations are going to be in more and more and more cells, and it's more difficult for your body to compensate for those mutations. And so generally that's seen in children, and the children often pass away because of these issues. Chronic leukemia is more prevalent in older people. Generally, these are mutations in cells that are much farther down the line, right? If only the eosinophils are containing this particular mutation, then some of the other cells might be able to compensate for that problem. And I leave you today with an animation of macrophages, which are a type of white blood cells, which are discovering bacteria that have invaded the body. They essentially take in those bacteria inside the cell, and they need to alert other white blood cells. And so they are going to immediately send out cytokines, and these cytokines are going to lead to the creation of more white blood cells, and they're going to call other white blood cells to the scene.